Thanks, babe.
The first bacteria on our list is going to be anthrax. Well, technically Bacillus anthracis. Bacillus anthracis normally lives in the soil. You can either inhale the spores. If you have like a cut on your skin or some other like opening in your skin and you touch something that has anthrax spores on it, you can also get the spores into your skin, and that will cause cutaneous anthrax. And then once these spores are inside of your body, if they're in the spore form, they'll break out into the functional bacteria form and they'll start producing toxins. And we'll kind of get into what those toxins do to your immune system and your body. Now, before you guys get all freaked out, how common is it to get anthrax? So in the US, there was like only a couple cases, I think, in the past year. And then in 2019, for example, the EU reported only 5 cases. So it's extremely rare to get, or not really extremely rare, but you know, it's not very common. And then globally, there's about 20,000 to 100,000 people per year that get it, which is a pretty broad range. And then I put this map here, which was published from a paper in 2019 that kind of shows with the black dots where all of the cases of anthrax were for that year. Canada showed up as a hotspot with 401 to 1,000 cases. And this actually... This is looking at the years 2005 to 2016. So there's some regions in Canada. So if you live in Canada, be careful. We see some cases in Russia, though not as much, in China, and then Australia, some parts of South America, Africa. Moving on, let's take a closer look at what this bacterium looks like. So if you were to go and do a gram stain, so a gram stain is where you take crystal violet and some other little dyes and you put it on your bacteria sample. And if your bacteria is gram positive, which means it has a very thick kind of like cell coating made out of peptidoglycan, it'll turn purple. So in this upper picture here, we have a, you see these like purple rod-shaped bacteria cells that are like all kind of stuck together. This is what Bacillus anthracis looks like under the microscope. And if we look down at the lower portion of the slide here, we can see kind of a cutaway, a computer rendering of kind of all of the different parts of the cell. And you'll notice right away that there's this blue shiny coating around Bacillus anthracis, and this is its capsule. And the capsule is actually extremely important for helping this bacteria evade your immune system, helping it to survive in the host. So the little like oval circular thing in the middle is an endospore, and this is the part that survives. Like let's say the bacteria is under stress, it'll just live in its little tiny endospore. And live on. Okay, so let's talk about the defense system that this bacteria has. So it has this capsule, which it's called a poly-D-gamma glutamic acid capsule. It's encoded by a specific plasmid. Now, inside of bacteria, they do have DNA like normal cells, but they're stored in these like, you know how DNA in humans is kind of long and like linear? In bacteria, they have their DNA in these like little circles. Usually it's all coiled up, but they're supposed to be circular. And there's these things called pathogenicity islands, and this is where bacteria store all of the genetic information that lets them make things like the special capsule that helps them evade the immune system. So this one is stored on PXO2, you don't need to remember that, and it's just a little tiny bit of DNA. This capsule, basically, it helps the B. anthracis escape neutrophil detection. So neutrophils are like these immune cells that will generally recognize bacteria based on their peptidoglycan wall, but you can see that the peptidoglycan wall is covered by a capsule, and so the neutrophil cannot see this bacterium. And so then there's another toxin that is encoded by one of these pathogenicity islands, and it's called lethal toxin. And this lethal toxin actually will go in and it'll kill macrophages. And so macrophages are another one of the main cells that normally will eat these bacteria. B. anthracis will produce 3 different types of exotoxins. One is the edema factor, which will cause elevation of intracellular C-amp. So C-amp is one of the cellular messengers, and that'll lead to a decrease of chloride ions in like the infected cell. This could be like a regular cell in your body. And then all of the water from inside of that cell will leak out and it'll cause edema, which is just swelling. There's also the lethal toxin, which I just mentioned. And this also causes tissue necrosis. And I'm going to show you guys some lovely pictures of tissue necrosis. Don't worry, they're not super gross. And then there's something else called protective antigen. Protective antigen basically will kind of bind to the other toxins and allow them to enter into host cells, like, you know, the cells in your body or something, and, you know, cause that tissue necrosis. So when B. anthracis enters into a host like an animal or a human, it uses iron to survive. The way that it gets iron is by using siderophores, and siderophores are like these little proteins that the bacteria makes. There we go. So siderophores will kind of they'll be released by the bacteria, and they'll pool in iron so that the bacteria can use that to power itself. Basically, one of the big sources of iron inside of the body are in blood. So in the heme molecule, and so these siderophores will kind of take the iron from the heme and transport it back into the B. anthracis bacterium. Right, so the lethal toxin kills the macrophages by phosphorylating or binding to, basically like interacting with a protein called P38, and this protein will then go and it'll be inhibited. So it won't, normally P38 will activate the macrophage to like produce different cytokines and different things that tell the macrophage to go kill the bacteria. But when the bacteria releases its toxin, it shuts down the macrophage's function, and then the macrophage actually ends up dying because it gets an opposing signal that tells it to kill itself. And so you get a lot of macrophage cell death. That's that. Now we're going to look at kind of the lab diagnosis. Like, how do you know if you have Bacillus anthracis? So you could do this by swabbing, taking a sample of the, sputum or like the skin, and you can take that sample and grow it on different types of agar. You can grow it on sheep blood agar. The reason you would grow it on sheep blood agar, and this is just a picture down here of some lovely white colonies of B. anthracis, is because you want to see if it's actually lysing the blood. Now, B. anthracis is related to Bacillus cereus, which is another pathogenic bacteria. It's also found in the soil, and so it could kind of be in the same place as B. anthracis. And B. cereus is known for lysing blood. So over here on the right, you can see that B. anthracis, there's no blood lysis, but then over here you see like around the B. cereus colonies, you see kind of like a little halo, and that's because all of the blood has been lysed. So that's one test to kind of differentiate between, is this anthrax or is it B. cereus? Another test that you can do is you can grow it on specialized media. There's something called PLET agar, and PLET just stands for polymyxin lysozyme EDTA thallus acetate. And thallus acetate is actually extremely toxic to humans. So people don't really like to make this type of medium, and so scientists have made other types of medium, but this is like the classic one. And basically only B. anthracis can grow on it. So it's very selective. The next thing you can do is you can stain for the capsule, and you can use this thing called McFadyen capsule stain. And in this image here, you'll see some green cells, which I am assuming are maybe red blood cells or something else. I don't know why they're green. This McFadyen capsule stain has different dyes than like the H&E stain that we talked about earlier. But you'll see that there's this little pink halo around the purple rod. So you can do a capsule stain. You can also do a gamma phage lysis test. They look like this, a bacteriophage. And they're like little tiny viruses that will kill bacteria. They look super metal. They have like these little claws that latch onto the bacteria. And every bacteria has its own special phage that it's susceptible to. And there's a certain one that's called gamma phage. There's like a whole other extra long name for it. But if you plate it on top of a field of B. anthracis growing on like a blood agar plate, and that phage is able to kill the bacteria cells, you'll see like little holes where the bacteria are now dead, which is super cool. There's other tests, but we won't go into it. There's also biosensors, which are really cool. So you guys probably have heard about anthrax being used as like a bioterrorism tool, weapon thing. And so because of that, governments funded projects to make biosensors that pick up tiny little amounts of anthrax spores in the air. And they do this by recognizing like either the protein, like certain types of like surface proteins on the B. anthracis, or they also use antibodies that are specific for certain parts of this bacterium. And then basically if there's like spores that are picked up by the biosensor, it'll transmit that into like a signal, and then it'll send that signal to like the operator or whatever. Finally, my favorite part, and I kept the pictures really PG, but there's some really horrible pictures out there if you look up anthrax and what it does to your skin. If you have cutaneous anthrax, it'll basically create these lesions due to that lethal toxin that's causing necrosis in your tissue. It's just killing all the cells, and it just leaves like this black hole of dead tissue. So symptoms could take 1 to 7 days to manifest. I heard somewhere that they could take even up to 60 days after first initially coming in contact with the spore to manifest. And then you can get swelling caused from that edema. And then these little ulcers, which they say is painless, but you will get a lot of them and it's not fun. There's other types of anthrax that you can get. Inhalation of anthrax is highly fatal. And there have been some public incidents of that over the course of the history of anthrax that we'll talk about in a little bit. There's gastrointestinal anthrax, so if you swallow the spore, that's also extremely fatal. These two are very rare. Most people, if they contract anthrax, it's through like the cutaneous, maybe it enters the skin through like some small cut or something. And then there's injection anthrax, which is also pretty lethal. And then finally, going away from that, we're going to talk about treatment and prevention, so you don't feel so scared. So if you do contract anthrax somehow mysteriously in your lifetime, which I hope you don't, you can take any sort of, you can go to your doctor, obviously, and they will probably prescribe you a beta-lactam antibiotic. The way that beta-lactam works is it binds to this enzyme that all bacteria that make a peptidoglycan wall have, and it's called transpeptidase. And when the beta-lactam antibiotic binds to the transpeptidase, it prevents the bacteria from making its cell wall. And this obviously, without a cell wall, the bacteria is just gonna like explode and die. And that's how the antibiotic works. The antitoxin is an antibody that binds specifically to the protective antigen. So remember that Bacillus anthracis codes 3 different types of toxins. One is the protective antigen, one is edema factor, and one is lethal factor. So in this little diagram here, we have this little purple oval, and that's the protective antigen. It lets the lethal factor and edema factor enter into the cell. Will give that antibody that kind of neutralizes and blocks protective antigen to protect you. Okay, hang on. This is a very wordy document, and you can tell that it was scanned because this was back in the day before they had PDFs. Okay, it's a lot of useless words. The most effective biocide was a 5% solution of formaldehyde in seawater. Formaldehyde is extremely reactive and reacts with protein and DNA and RNA, because DNA and RNA are composed of similar things as protein. With protein, it produces intramolecular crosslinks, and if you're curious here, I'll highlight it. It is said to be sporicidal because it penetrates the interior of the spore, because remember, it has that really tough spore, where it presumably inactivates nucleic acid. There are some spores that will resist the action of formaldehyde for some hours, but our experience on Grunard Island showed that a 7-day exposure effectively inactivated all bacterial spores present in the soil. It is possible that the 40-year residence of the spores in the soil may have made them more sensitive to the effects of biocides, but this is uncertain.